In prokaryotes like bacteria, DNA replication occurs before cell division, ensuring that each daughter cell receives an identical copy of the genetic material. The process begins at a specific point called the origin of replication on the circular DNA. At the origin, the enzyme helicase unwinds the double-stranded DNA, breaking hydrogen bonds between the base pairs. This creates a replication fork, where each strand is now free to serve as a template for the new DNA. Next, primase synthesizes a short RNA primer, providing a starting point for DNA polymerase III. On the leading strand, DNA polymerase moves continuously in the 5' to 3' direction, adding complementary nucleotides to the growing strand. On the opposite strand, called the lagging strand, DNA synthesis occurs in short segments. Primase adds primers at intervals, and DNA polymerase III synthesizes Okazaki fragments between these primers. Once the Okazaki fragments are synthesized, DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase seals the gaps, connecting the fragments to create a continuous lagging strand. Replication continues until the two replication forks meet at specific termination sites. The result is two identical circular DNA molecules, each ready to be passed on to a new daughter cell during cell division. In summary, prokaryotic DNA replication is a well-coordinated process involving enzymes like helicase, DNA polymerase, and ligase to ensure accurate duplication of the bacterial genome. This ensures each new cell inherits an exact copy of the DNA. Transitions and visual techniques are crucial for clarity. Use fades between each major step to provide smooth visual transitions. Zoom in for detailed actions like helicase unwinding or DNA polymerase adding nucleotides, then zoom out to show the overall DNA structure and movement of replication forks. Motion graphics, such as arrows, glowing paths, or highlights, can effectively show enzyme movements, strand synthesis direction, and interactions between components. For replication fork movement and strand synthesis, maintain a consistent motion direction to help viewers follow the process logically. Enhancements can make the learning experience more engaging. Use color differentiation to distinguish between original and newly synthesized strands, as well as enzymes like helicase, primase, and polymerase. Simple icons, such as a scissor icon for helicase or a wrench icon for ligase, can represent different enzymes. Adding a subtle pulse effect on important elements like the RNA primer or Okazaki fragments can draw attention. Finally, include on-screen labels for key components and processes such as the replication fork, DNA polymerase, and the leading and lagging strands. Let's outline the storyboard. Scene 1, the bacterial cell is shown. Zoom in on circular DNA, focusing on the origin of replication. Scene 2, helicase unwinds the DNA strands, creating a replication fork. SSB proteins bind. Scene 3, primase lays down RNA primer. DNA polymerase III synthesizes the leading strand continuously. Scene 4, primase adds primers at intervals on the lagging strand. DNA polymerase III makes Okazaki fragments. Scene 5, DNA polymerase I replaces RNA primers with DNA. DNA ligase seals Okazaki fragments. Scene 6, zoom out, show termination of replication, two circular chromosomes separate.